Good morning, my dear students. Welcome to my channel Patel Science Academy. As you know, this is your Shahza sir, and you are watching right now Patel Science Academy. Dear students, I welcome you in the chapter Molecular Basis of Inheritance. Dear students, in this chapter, today you will learn about the DNA fingerprinting, the topic from the genetic engineering. So, dear students, what is genetic engineering and what is DNA fingerprinting. Before we go in the depth of this topic, I would like to explain you about the DNA. What is DNA? DNA is a genetic material uh, which is present on the chromosome, and this is the unique identity of every living organism. So actually the genes present on the chromosomes are responsible for determining the characteristics of the organism as well as for the inheritance of the characteristics. So dear students, due to the recombination of the paternal and the maternal genes, we differ from our parents. That's why the reason is only maternal and paternal genes, and genes makes DNA and DNA makes chromosomes. So differences also arises due to the infrequent mutation that occurs during the gamete formation, that is during the cell division. So due to all these factors, every individual has its unique genetic makeup, and which may be called as fingerprint. So dear students, that genetic makeup is the fingerprint of every living organisms, and the technique developed to identify a person with the help of a DNA restriction analysis, that technique is called DNA profiling or DNA fingerprinting, in which every person's genetic makeup is identified by this technique, which is called DNA fingerprinting or DNA profiling. The technique of DNA fingerprinting was first given by the British geneticist Dr. Alec Jeffrey in 1984. So dear students, we'll see about this DNA profiling or DNA fingerprinting. Its detail, a technique used to distinguish between the individuals of the same species using only sample of their DNA. So dear students, by this technique we can distinguish between the individuals of the same species only by just taking their DNA sample. Two humans will have the vast majority of their DNA sequence in common. The genetic fingerprints exploits highly variable repeating sequence that is called mini satellite. So I'll explain about that mini satellite in the next slides. The two unrelated humans will be likely to have the different numbers of mini satellites at a given point. So it is all about the DNA material, that is genetic material, and the mini satellite. So dear students, we'll see about it in the next slide. So look at this man. This is the person known as Alec Jeffrey. This is Alec Jeffrey. He invented the process of DNA fingerprinting in 1985. So this is that man. Dear students, what are the applications of DNA fingerprinting? DNA fingerprinting, it is very useful. Dear students, it is very useful in the crime scenes. Actually, in a forensic science, DNA fingerprinting is used to solve the problem of the rape and some complicated murder cases. Then human relatedness, DNA fingerprinting is also used to find out the biological father or mother or both of the child in case of the disputed parentage. And the DNA fingerprinting is used in a pedigree analysis in a cat, dogs, horses, and humans too. So anthropology studies disease causing organisms, diagnosis and their cure, food identification, human remains, and monitoring the transplants. All these are the applicable aspects of DNA fingerprinting or DNA profiling. So this is very useful technique for living organism. DNA fingerprintings. In the DNA fingerprinting, dear students, in the DNA fingerprinting, there is one term that is called VNTR. What is VNTR? The full form of the VNTR is variable number of tandem repeats. So what is this variable number of tandem repeats, and what are the use of this variable number of tandem repeats? This is most important thing in the DNA fingerprinting. So dear students, DNA fingerprint technique is based on the identification of the nucleotide sequence present in this wonder molecules. About 99.9% of nucleotide sequence in all persons is same. Only some short sequences of nucleotide differ from person to person. So in a population, every person shows unusual sequence of that 20 to 100 base pairs, and these 20 to 100 base pairs are repeated several times. So these 20 to 100 base pairs which are repeated several times, these are termed as a VNTR, that is called variable number of tandem repeats, VNTR. The length of the region having the VNTR is different in each individuals, and on the basis of it, this VNTR actually its on the basis of its length, because this is the key factor in this DNA profiling, the length of this VNTR, because it is different, it is having the different length in the different living organism, and we have to just check this VNTR length in the different living organism to know about their DNA profile. So there are different steps which are involved in the DNA fingerprintings, and that we'll see one by one. So I hope you have understood VNTRs. So in the body there are organ. In the organ are made up of tissue. Tissue is nothing but a group of cell. In a cell there is nucleus. In the nucleus there is a genetic material called chromosome. In the chromosome there is a genetic material, there is a DNA, and in the DNA there is a gene, and in the gene there is a 22, there are nucleotides, the sequence of nucleotides, and that 20 to 100 base pairing of nucleotide, base pairing in the nucleotide is called VNTR. And in the DNA fingerprinting, we just know about the VNTR, we just identify the VNTR. So once some have chromosomes, a short sequence of the DNA has been repeated a number of times. That repeated number may vary from 1 to 30 repeats. These repeats regions are usually bounded by the specific restriction enzyme sites. Cut out the segments of the chromosomes containing this variable number of tandem repeats and identify the VNTR for the DNA sequence of the sequence of that repeat. So dear students, this is about this VNTR. You have to just know about the VNTR, and you have to just identify the VNTR for the DNA of that particular person. So dear students, variable number of tandem repeats means the VNTR, the sequences that are repeated multiple times, which is 20 to 100 in number, and the number of repeats varies from person to person. So we'll just see here the example of one VNTR in this sequence. So this is one of the gene sequence, and we'll see here 5 repeats are there in the as a VNTR. So you can see here GA, guanine, adenine, thymine, cytosine. This is the sequence. Again GATC, again GATC. So these sequence is repeating itself multiple times. GATC, then GATC. So dear students, this GATC is the base pair guanine, adenine, thymine, and cytosine. So this base pairing are repeating itself one time, two time, third time, fourth time, and fifth time. So So because this sequence has repeated itself 5 times, so we can consider it one VNTR, because it is the sequence which repeats multiple time in the uh, gene. And in the second example we'll see here GATC 2 time it is repeating, that's why we can consider it one of the VNTR. So this is called variable number of tandem repeats, and in the DNA profiling or DNA fingerprinting we have to just identify this sequence there. So this is about the VNTR that we always identify in the DNA profilings. Dear students, there are different stages. Stages includes the isolation of the DNA, the restriction digestion, gel electrophoresis, then southern blotting test, then selection of the DNA probe, and hybridization and photography. All these are the 7 steps of uh, DNA fingerprinting. So dear students, will see all these steps one by one: the specimen collection, DNA extraction, PCR technique if necessary, then cutting the DNA with the restriction enzymes to get the restriction site, then gel electrophoresis, then southern blotting test, then adding of the radioactive or fluorescent probe, and the X-ray, and the photography by the X-ray or develop or look at the under ultraviolet light. So these are different stages of DNA fingerprinting, and we have to just be familiar with all these steps one by one. So first step is isolation of DNA. The DNA must be recorded from the cells or the tissue of the body host, and only small amount of tissue like the blood, semen, saliva, urine, hair, teeth, bones, and tissue are needed to identify the DNA of that particular person. So dear students, we can take the blood sample for the DNA fingerprinting, or semen in the male, saliva, urine, hair sample, teeth, bones, and tissue. We can take anything: blood, sperm or semen, teeth, tissue, urine, hair sample, bones or muscles, any tissue, anything we can take from this, and saliva also. So this is about the sample we have to collect for the DNA profiling from the patient, or from the person going to be checked. So dear students, this is first step. We have to isolate simply this DNA sample from that person. So stage 1, the cells are broken down to release the DNA. This is that step number 1, and if only a small amount of the DNA is available, it can be amplified using the technique PCR, that is polymerase chain reaction. Dear students, by this technique is necessary only if we have collected and we got a very small amount of the sample. If we have sufficient amount of the sample, there is no need to go for the PCR, and if we don't have the sufficient amount of DNA sample, so we can go for this technique to get the multiple copy of that sample. So stage 1 is simply the collection of the sample or isolation of the sample in which cells are broken down to release the DNA. Thereafter stage 2 is the DNA. In the stage 2, the DNA is cut into the fragments using the restriction enzyme. So this is important technique in which the restriction enzyme is used, and it is applied on the DNA sample to get its small fragment. So each restriction enzymes, it cuts the DNA molecule at a specific base sequence. So as you can see here in this example, if we cut the restriction enzymes, if we cut this DNA samples and we apply here this restriction enzyme, so it cut the enzymes from this it cut the DNA sample from this AATT from this sequence. So this enzymes is applied there, which is called restriction enzymes, and the isolation of the DNA is treated with the restriction enzymes, and the restriction enzymes cut the DNA into small fragments having the variable length, and this phenomenon is called RFLP, that is called restriction fragment length polymorphism. Restriction fragment length polymorphism. Second stage is the isolation of fragmentation of this DNA molecule. So dear students, in the first step we have isolated, we have collected the sample, and in the next stage we have applied the restriction enzymes to cut the DNA molecule into a small fragment, into small pieces by applying the restriction DNA enzymes, and this process is called RFLP, that is called restriction fragment length polymorphism. Thereafter, the third stage include the gel electrophoresis, and in this stage the DNA sample are loaded for the agarose gel electrophoresis under an electric influence, and their students, the DNA fragments which are negatively charged, remember this thing, DNA fragment is negatively charged, it moves towards the positive pole on this electrogel plate, and the movement of this fragment depends on length of the fragments, and this result into the formation of the bands, because DNA molecules moves towards the positive charge, because it is DNA is having negative charge. So because of moving of that DNA, bands appears there, and because of that bands, this dsDNA splits into ssDNA, double stranded DNA. It splits or break into single stranded DNA by alkali treatment. So this is the third step, in which the fragments are separated on the basis of size using a process called gel electrophoresis, and DNA fragments are injected into the vial, and electric current is applied along the gel so that it can take the shape of the bands. So you can see here the DNA of different size, and this is the power source, this is anode, and this is cathode, positive and negative charge, and agarose gel is applied there, and DNA is negatively charged, it moves towards the positively charged, and bands are formed. This is stage number third. So as you know, DNA is a negatively charged, so it is attracted to the positive end of this gel, and the shorter DNA fragments move faster than the longer fragments, as you can see here in this gel. So these are longest DNA, and it is moving slowly, and small DNA, it moves faster as compared to long DNA, because of this agarose gel, and because they move towards the positive charge. So these are negatively charged DNA, it is moving towards the positively charged DNA, and because it moves, so these bands are formed there, and we have to just identify these bands, because we are identifying here this DNA molecule. Stage 4, it includes the pattern of the fragment distribution is then analyzed, and in this step 4, the southern blotting, the separated DNA fragments are transferred to a nylon membrane or a nitrocellulose filter paper. Now this fragment band DNA, it is applied or it is transferred on the nylon membrane, or either on the nylon membrane or nitrocellulose filter paper. It is applying on the filter paper or membrane by placing it over the gel and soaking them with filter paper overnight. So this is called southern blotting test. The southern blotting was named after the Edward M. Southern, who developed this procedure. And dear students, to oversimplify, the DNA molecules are transferred from an agarose onto a membrane, and southern blotting is designed to locate a particular sequence of the DNA within a complex mixture. So simply DNA southern blotting means the separated DNA fragments are transferred to a membrane, nylon membrane or a filter paper, nitrocellulose filter paper, by placing it over the gel and it soaks and because we kept it for overnight. So this is step number 4, southern blotting test. A principal procedure and the application. It includes the extract and purify the DNA from the cells. DNA is restricted to the enzymes separated by the electrophoresis gel. Denature the DNA, transport to the nitrocellulose paper or blotting for the southern blotting. And then next stage is add the label probe for the hybridization to take the place, then wash up the unbound probe and autograph. These are the different stages of DNA profiling. So we are watching here all these steps. As in the first step we have seen DNA restriction enzyme is applied here. DNA, as in the first stage we have seen here, DNA restriction enzyme is applied here, and because of that enzymes, DNA converts into this small fragment. What happened in the next stage? In the next stage, this is applied. This is applied for the southern blotting test, because of which this is applied for the electrophoresis, because of which we got band, and then we have put it for the next stage, that is southern blotting test. Likewise, in which it is this DNA separated fragment are transported on the membrane, and it soaks the gel and filter paper with the filter paper overnight. And then what happened, it is then for the it goes for the next stage, that is called selection of the DNA probe. Known sequence of the single stranded DNA is prepared, that is called probe. The single stranded DNA whose sequence is known to us, so that is called probe. And it is called DNA probe because we know all the sequences of that DNA. So we are applying that we apply that DNA probe, and this probe is obtained from an organism or prepared by the cDNA preparation method. The DNA probe is labeled with the radioactive isotopes. So dear students, by comparing with this radioactive isotopes, we can go for the next stage. So what is next stage? Next stage is the DNA fingerprint hybridization. So because in the fifth stage, probe is applied there on the single stranded band of the DNA after the southern blotting test. So as you know the sequence of the DNA probe, and if we are applying it on that particular sequence on that particular sample, so what will happen? The final DNA fingerprint is built by using the several probes, and it resembles the barcode. So you can see here different barcodes appears here, and it has now become double stranded. So DNA probe is added to the nitrocellulose filter paper containing the host DNA, and the, as you know, probe DNA whose sequence are known to us. We know the sequence of that DNA, and we have applied that DNA probe on the sample DNA, and the single stranded DNA probe pairs with the complementary base sequences of the host DNA strand, and in this way, as a result, DNA hybrids are formed on the nitrocellulose filter paper, and remaining single stranded probe fragments are washed off whose sequence are known to us. So in this way we can go for the identification of the DNA sample, and it is all about the probe, because if as for example on the probe first sequence is A, so as you know A is adenine, and then second number is guanine, AG. So if on the probe A is there at first number, so definitely A will bind with T, thymine. So if thymine will be there somewhere on that host DNA, so it will pair with that T, otherwise it will not pair. And G will pair with C, because it is the natural process, G pairs with C. guanine pair with cytosine. So if cytosine will be there somewhere in that host DNA, so it will make either pair with that, otherwise it will not pair with that sample DNA. So likewise, for the multiple use of the DNA probe, we can get the DNA record of that host DNA. So likewise, these are the samples, collection of the sample, then it's what we say, isolation, then restriction, then gel electrophoresis, then southern blotting, selection of DNA probe and its application, its use, and then hybridization, and finally after getting it, the next stage is this one, the photography. In the photography, the nitrocellulose filter paper is photographed on an X-ray film by auto radiography, and the film is analyzed to determine the presence of hybrid DNA. So likewise, this DNA is identified by a person. So dear students, there are some famous cases identified by the DNA fingerprinting technique. So Colin Pitchfork was the first criminal. Colin Pitchfork was the first criminal caught by this technique, but on the basis of this DNA fingerprinting evidence, and he was arrested in 1986 for the rape and murder of two girls and was sentenced in 1988. Thereafter, the father of genetics, as you know, the father of genetics is Professor Lalji Singh. Band carrot snake, Y chromosome of the female, X Y in the female, YY in the male. These are called BKM, that is banded carrot mini satellite chromosome, was identified by this man, and he has collected that sample. So this is about this DNA fingerprinting. Dear students, thank you very much for watching this video, and please do subscribe my channel, share with your friends, and like it, and press the bell icons to receive my instant notification of the new video. And dear students, you can apply, you can go for the online test of this based on this lecture. For that online test, just go in the description box in this lecture, below in the description box, and click on the link and start solving the MCQ online test. And just after the completion of the test, you can see your score. So you can give the test, you can attend the test too on the basis of this DNA fingerprinting in this lecture by going in the description box. Thank you very much for watching this video.